Welcome back, students. In the previous section, we saw how the lac operon is being regulated by the levels of lactose and by the levels of glucose. Now we saw that when lactose is present, it binds to the repressor protein and allows the transcription of the genes to take place so that more and more lactose can be metabolized. But there may be situations where what is happening when only lactose is present and glucose is absent. One more mechanism is taking place. Till now, whatever I had told in the last section is enough to know about the basics of lac operon. But if somebody is interested in knowing more about this lac operon, I would like to suggest a bit more in the details regarding this. So a bit more detail. What is happening is another mechanism is also taking place when this lactose is present and glucose is absent. So in the absence of glucose, adenylyl cyclase is active. Absence of glucose, no glucose. Adenylate cyclase is active. So what? It increases the cyclic AMP levels. So what? When cyclic AMP levels increase, this cyclic AMP will be bound by one more protein. This cyclic AMP will bind with another protein. Okay. Now this protein will go and bind to the catalytic CAP site. The CAP site which is there, it will go and bind there. So if it binds there, what will happen is this RNA polymerase, which is supposed to polymerize and cause transcription, that will become more efficient. So whenever glucose is absent, we need this transcription to take place. So when glucose is absent, when you want transcription to take place and you want it at a higher efficiency, adenylate cyclase will form cyclic AMP, cap protein, cap binding site, it will go and bind and increase the efficiency. So cyclic AMP binds with the cap. This cyclic AMP cap complex binds to the cap site. This causes RNA polymerase to efficiently initiate transcription. So this maximizes the expression. So this is known as positive regulation. So we'll look at one more situation when both glucose and lactose are present. Glucose is also there, lactose is also there. Which is the preferred fuel? The preferred fuel is glucose. So when the preferred fuel is glucose and lactose is also there, this lactose will form all lactose and it will cause the binding of the repressor protein. So there must be some mechanism by which when lactose is also there, glucose is also there, this gene should be switched off. This gene should not transcribe. So what will happen when both glucose and lactose are present? So in the presence of few molecules of permease results in the uptake and conversion of lactose to all lactose. So all lactose binds to its receptor. So RNA polymerase should be able to transcribe, right? There is no binding out here. So it should be able to transcribe. But E. coli has glucose. So why do that transcription unnecessarily? It is already having glucose. It wants to metabolize glucose. Why transcribe lactose metabolizing enzyme? So here comes the role of cap again. So in the presence of glucose, there is no cyclic AMP. This cyclic AMP is not there. So if cyclic AMP is not there, what will happen? Cap cannot go and bind here. So this binding is gone if there is glucose. When cap cannot bind, what will happen? RNA polymerase cannot initiate transcription. When RNA polymerase cannot initiate transcription, this formation of mRNA does not take place. So this type of mechanism in which when a metabolite is there, this metabolite is cap protein cannot go and bind here. What do you call this? We call this as a type of inhibition that is taking place of glucose by glucose of this expression. So when we look at the whole summary, when glucose is absent, lactose is present, what is happening? All lactose is formed. So what binds to repressor? So what cannot bind to the operator site. So RNA polymerase can start transcription. When glucose is absent, adenylate cyclase is active and this will increase the cyclic AMP and cyclic AMP binds to cap and this will bind to the cap site and stabilizes RNA polymerase and thereby the protein product is formed. This is the whole summary that you need to know. The extra ones what I have told to you is an extra thing that I have told. Now there is one more operon, and that is known as the tryptophan operon. MCQ point of view, can it be asked? Yes, but it is mainly related to anabolic pathways. Again, this is in prokaryotes. Now I'll just tell you, the tryptophan operon codes for 5 proteins that are required for the synthesis of tryptophan. Now what we saw, this pathway was a catabolic pathway where lactose was going to be degraded. Now what I am talking is an anabolic pathway where tryptophan is going to be synthesized. So when tryptophan is present, the tryptophan binds to the repressor, and this complex binds to the operator, prevents transcription of genes and negative control. The another mechanism is attenuation. Transcription is initiated, but it is terminated before the completion of the hairpin structure by the formation of a hairpin structure at the 5' end. Since in prokaryotes, transcription and translation are a coupled process, attenuation results in the formation of a truncated non-functional protein. You need not know more than this regarding the tryptophan operon complex. This much is enough. Okay. So let us look at one thing. Is gene expression regulated only at the level of transcription? Only can you regulate at the level of transcription? No, actually. So regulation occurs at the level of translation also and at the level of rRNA also. So this helps the microbe to adapt to different environmental stresses, and this is known as the stringent response. And there are what are called as regulatory ribosomal proteins which are involved in all this. All this till now what I told was regarding prokaryotes, both the lac operon and the tryptophan operon. But what is how are genes regulated in eukaryotes, including human beings? How is this happening? So let us go for regulation of gene expression in eukaryotes. In eukaryotes, are there any operons? Are there any structural genes? Are there any regulatory genes? So let us look at all this. Now, eukaryotic gene expression is very complex. Is it simple? No, not as simple as that happened in the bacteria. Remember, in the bacteria we had a circular DNA. It is easy to regulate. Now, what is the problem with eukaryotes? We all have a nucleus. The nucleus has a nuclear membrane, and that brings about a lot of complications. Okay, and we can also have a wider range of regulatory mechanisms. So in eukaryotes, regulation occurs at multiple levels. It occurs at the level of transcription. It occurs at the level of mRNA editing. It occurs at the level of alternative mRNA splicing can take place. It can occur at the level of controlling the mRNA stability, control the translational efficiency, protein stability, activity, degradation. At different levels, we have got different ways we can regulate the whole product, the functional product of a gene. So let us look at the different levels. Transcription, post-transcriptional modification, mRNA splicing, mRNA stability, control of translational efficiency, protein stability, activation and targeting. Let us look at all of these methods. So why can't we have something as good as a lac operon that was happening in the bacteria? So a simple system like lac operon or tryptophan operon with an activator or repressor would not allow the flexibility that is needed in eukaryotic cells. A lot of flexibility is required. There is simply not enough room in the. You remember we have a huge DNA and this is all put into a small nucleus. So this much huge DNA to regulate plus one more repressor protein, activator protein or promoter site, operator site, the DNA could have increased, and there is all these activator proteins to come and associate with the DNA in the vicinity of promoter. There is no space. So many conditions must be true for a cell to begin synthesizing a specific protein. If A is true, B also should be true, C also should be true. Only then this protein is going to be synthesized in a eukaryotic cell. Simple, only one condition. If A is present, only then this protein is going to form is not sufficient for us. We need multiple regulatory systems to act. Only then that protein has to be formed. Only then that gene has to be turned on. Okay. So let us look at the different subtopics under which I will be talking about regulation of gene expression in eukaryotes. I'll talk about the transacting elements, The cis-acting elements, in which I talk about intracellular receptors and the cell surface receptors, regulation through modifications of DNA, and the post-transcriptional processes. So let us look first at the transacting molecules. These are the transcriptional activators. They facilitate transcription initiation complex. What are transacting molecules? Are always products of some other gene. They are proteins. They help in the formation or regulation of this gene. They facilitate formation of transcription initiation complex. Now, regulation is achieved by the formation of a multi-protein complex bound to the DNA with protein and protein DNA interaction. So this may be a protein-protein interaction or a protein-DNA interaction, which will help in regulating the whole gene. This is known as combinatorial control, because both the protein and the DNA are required, or a protein-protein complex should form first, and this protein-protein complex will go and bind to the DNA and form a protein-DNA complex. So a lot of things are involved in this. This is called as a combinatorial control. Many times such transacting elements, they even fold the DNA. A lot of all these proteins will come, a bigger complex is formed, and the transacting elements will fold the DNA which is present, a double-stranded DNA that is present is folded, and then it brings about gene regulation. So such things can also happen in eukaryotes. Transcription can be inhibited also. Okay. Now there are enhancers. What are enhancers? Whenever we say enhancer, there are special cis-acting DNA sequences that increase the rate of initiation of transcription by RNA polymerase II. Enhancers can be upstream or downstream. They can be close or nearly 1,000 base pairs away from the transcription site. They can be on either strand of the DNA. They need not be specific to one strand of the DNA. And the enhancer activator complex can interact with the transcription factor at a gene's promoter and allow transcription to begin. So there may be a formation of an enhancer activator complex. I have the transcription initiation site. You have this big DNA. If this is over here, this may be multiple. This cis-acting element may be somewhere here. It may be very far, or it may be close, or it may be acting on some way, and it will cause all this They can be on either side of the DNA, either strand of the DNA, either here or there. But basically, when we say cis-acting, it means they are present on the same DNA. Okay, the enhancer can interact with the transcription factor at the gene's promoter and allow transcription to begin. DNA looping is often involved. Even in this, for this, if it is so far away, the whole thing may be looped by transacting, cis-acting, come together and regulate the gene. Now, there are what are called as cis-acting regulatory elements. Now, to coordinate a group of genes, remember in eukaryotes, operons are not there. So, to coordinate a group of genes, we have cis-acting regulatory elements. So, a protein binds to the regulatory element for each of the gene, even if they are on different chromosomes. So, to genes, a group of genes may be there, and these may be on different chromosomes. They may not be on the same chromosome. Suppose we have one chromosome here, one chromosome there, but all this, I am just showing you. This is one chromosome with one gene. This is another chromosome with another gene. Another chromosome with another gene. Okay, one gene here, a group of genes. All these group of genes can be acted upon by one component itself. To coordinate this group of genes, we want all these gene products at the same time, or we don't want all these gene products at the same time. So a protein can bind to the regulatory element, maybe on different genes, maybe on the same gene, and regulate the activity of these genes, even if they are on the different chromosomes. They affect the expression of a group of genes. So most of the hormones regulate gene expression by this mechanism. All the hormones usually regulate the gene expression by this mechanism, because we want a group of genes, group of enzymes to be active. So this cis-acting DNA elements are called as hormone response elements, hormone response elements. Now the hormone response element, what does it do? It binds to transacting protein factors and regulates gene expression. So suppose there is a hormone, what will happen? This hormone will form a complex with its receptor. So this receptor may be from where did it come? It is a transacting element. So it formed a hormone receptor complex. What does this hormone receptor complex do? This hormone receptor complex can go and bind to a certain area of the DNA. It will go and bind to a certain area of the DNA and thereby regulate. This hormone receptor complex can go and bind to a certain area of the DNA and regulate gene expression of a gene that is present further ahead. So this is known as the action of hormone response element and the transacting protein factors. Now further to know about this, so if the hormone has an intracellular receptor, there may be this I was talking about binding directly. Okay, sometimes the hormone may have an intracellular receptor or it may have a receptor on the cell membrane. So if the hormone has an intracellular receptor, the signal transduction pathway is direct. Hormone plus receptor, hormone receptor complex. This will go and bind direct. There are no second messengers involved. Now hormone binds to its receptor, causes a conformational change in the receptor, activates it. This enters the nucleus, dimerizes, binds via a zinc finger motif to the DNA. It binds on the specific sites on the DNA which are called as hormone response element, which may be again present on different chromosomes. This allows coordinated expression of a group of target genes. The hormone response element could be an enhancer, and sometimes the HR complexes in association with co-repressor inhibit transcription. Sometimes it inhibits, sometimes it activates. If it is going to inhibit, it requires a co-repressor. Only then it can inhibit. So this is one mechanism where the hormone has crossed the cell membrane and bound to its receptor, brought about a conformational change so that it can go and bind to the DNA. Now there is which are the examples for this? Glucocorticoids, mineralocorticoids, vitamin D, thyroid hormone, all of these, retinoic acid, they all act upon in this manner. They all control gene expression and the formation of different proteins by this mechanism, acting at the level of the DNA. Now, other than that, are there any other ways? So, suppose the hormone is having the cell surface receptor. This is present in the nucleus and this. Suppose the hormone is having a cell surface receptor. What does it happen? So a hormone binds there. The hormone goes and binds to its receptor, and then what happens? It results in the formation of a second messenger. So for example, glucagon increases the cyclic AMP levels. So what? This leads to the activation of a transacting protein, cyclic AMP response element binding protein, or CREB. And this CREB then binds to DNA at the cyclic AMP response element and transcription is activated. So we have a second messenger being formed. This second messenger will be acting upon by another protein. It will combine with another protein, and this protein will now form this protein, cyclic AMP protein complex, cyclic AMP protein complex, cyclic AMP protein complex will now then go and bind to the DNA and regulate gene expression, and transcription is activated. This is all what I have told were cis-acting ways, regulation at the different DNA levels only. So are there any other processes? As I said, regulation occurs at different levels. One is at DNA level. It can also occur at the different levels. So regulation of post-transcriptional processes of mRNA. The splice site can be chosen. mRNA stability can be affected. We have something called as RNA interference and translation of mRNA. At all these levels, the gene expression can be regulated. So we saw how different hormones can bring about different regulation of by acting at the cis level. Remember, cis and trans means cis means usually from the gene, and it is the regulatory element is also present on the same gene. Trans means it requires a gene product which has come from a different gene product from a different chromosome or from a different DNA, and then this transcription has acted on it. So a different chromosome, so usually it may be a different site on the DNA actually, and these receptors have come from there, and it has caused the difference in the action. So let us look further upon the different ways what happens to this mRNA. So right now, till now we were talking at the level of the DNA and transcription level. Let us look at how regulation of gene expression occurs further for this mRNA or to the protein, how they can be affected in the next session. Thank you.